At the University of Missouri, Dr. Burke's lab is conducting research on the spread of HIV. The research focuses on the virus's replication process. The hope is that by inhibiting the virus's ability to replicate, the terrible disease will no longer be able to spread. The HIV virus causes a progressive failure of your immune system, allowing other infections and cancers to thrive. A virus is a very small organism that replicates only through living cells of other organisms. The HIV virus approaches a cell and interacts with its surface. HIV inserts its own RNA into the cell. Once the RNA is inserted, an enzyme, which is a special type of protein, binds to a certain spot on the virus's RNA and makes a DNA copy. Next, the shell begins to move towards the cell's nucleus, where the DNA is stored. Sometimes the virus's genes lay dormant for years before being activated. But when the virus's DNA is turned on, many copies of the RNA are made. The RNA leaves the nucleus with the information to make a new virus and moves towards the cell membrane. Once it reaches the cell membrane, a new virus starts to bud off. Many new viruses are made from an infected cell. These new viruses can infect new cells, and the cell cycle repeats.
But that first step of copying the RNA into the DNA, that's the part that we're targeting. There's a viral protein required for that, and we think we know pretty well how to mess up that protein so that it can't make the DNA copies. If it sits around being only RNA and never turns into DNA, you never have a successful infection. And that's our target. Cells are always making parts. They're making cellular parts, and those are molecules. And so we've designed a series of molecules that we can teach the cells to make. And as they make those molecules, they can then use them as defenses against the virus. They snuggle up next to the protein and bind onto it. They have just the right size and shape to bind to the protein and prevent that protein from doing its normal job. It's just like when you want to go to the store and you want to drive your car and you find your dog sitting in your driver's seat. As long as that dog's sitting there, you're not driving anywhere. So the difference between that example and what we're trying to do is our dog won't move.
Dr. Burke's lab is working to figure out how to introduce these molecules into infected cells with HIV. Our hope is to find the best avenue to deliver these small molecules that stop the infection so that our research can help the 33 million people living with HIV today.